Welcome to the Media Preparation Lab. Today we're going to prepare potato dextrose agar, a widely used medium for growing fungi and bacteria in the lab.
A granulated PDA. Yan, gusto ng PDA?
Potato dextrose agar.
Next, we add 1 liter of distilled water to the flask, stirring well to dissolve the ingredients. Now, we heat the mixture to 121 degrees Celsius for 15 minutes to pasteurize the medium and kill any bacteria that may be present.
Transfer.
After pasteurization, we allow the medium to cool slightly before pouring it into petri dishes or other sterile containers. And that's it! Our potato dextrose agar is now ready to use for cultivating fungi and bacteria in the lab. Today, we will guide you through the essential steps for decontaminating lab containers and petri plates, followed by proper sterilization using pressure sterilizer. Gather materials, you'll need the following. Cover the lab containers and petri plates using lab paper. Place the wrap items into lab cellophane. Set the pressure sterilizer to the appropriate setting.
Collecting diseases of different crops on different areas. Plant disease is defined as a state of local or systemic abnormal physiological functioning of a plant, resulting from the continuous prolonged irritation caused by phytopathogenic organisms. Cutting a box shape in the advancing portion of the tissue from mango fruit. Preparing for petri plates. 1 plate for 70% solution of alcohol and 3 for sterile distilled water. Dipping the cut specimen into alcohol or Zonrox bleach first. After dipping to alcohol, dip to each sterile distilled water. Same process with anthracnose, dip to 1 plate with alcohol and then 3 petri plates with SDW. Anthracnose or fusarium both undergo the final process, which is the planting of the cut specimen into the petri plates with PDA media. In bacteria, there is a technique known as dilution to reduce microbial load and widely separate the bacteria from other undesired microorganisms. Break the part where you want the bacteria to infect your carrot. Harvest the bacteria from a single colony in pure cultured media. Swab or paste it into the marked part of your carrot. Always flame sterilize your wire loaf. Then, repeat the same process. Here is an inoculation of Pictobacterium bacteria and petrichai. The microorganism should be recovered from the experimentally infected host and shown to be the same species as the one originally isolated from the diseased plant. This confirms that the microorganism recovered from the experimental host is indeed the cause of the disease. In re-isolation, all you're going to do is repeat the isolation and inoculation process. Koch postulates are a set of guidelines crucial for establishing a definitive link between a specific microorganism and a particular disease. They provide a framework for scientists to rigorously investigate and confirm the causative agent of an infectious disease, guiding research towards effective treatments and preventative measures. By outlining the necessary steps to isolate, cultivate, and reintroduce a suspected pathogen, Koch postulates have evolutionized our understanding of infectious diseases and continue to be a cornerstone of plant pathology.
Welcome! I'll be taking you along on our educational tour where we explored various farms and discovered valuable lessons along the way. Join me as I share the insight and knowledge we gain from this enriching experience. First up, in Trento Agri-Industries, Inc., Iho Resources Corporation, located in the heart of Agusan del Sur. Here, banana cultivation, especially the Cavendish variety, is not just livelihood, but a cornerstone of a region's agricultural economy. However, beneath the beauty of this expansive banana plantation lies an ongoing struggle, the fight against devastating banana diseases. Some of the most concerning are Panama disease caused by Fusarium oxysporum FSP cubensi, Moko disease, and the recently emerging bacterial cholera caused by Pectobacterium. We collected different specimens to isolate them in the laboratory to confirm the causal organism behind these diseases in the banana. These diseases are not just agricultural challenges, they're economic threats that affect farmers, workers, and entire communities. With the farm situated in a flood-prone area, water serves as a major conduit for spreading this harmful pathogen, complicating disease management effort. As we wrap up our visit here in Trento Agri-Industry Inc. in Agusan del Sur, we are now heading to our next stop in Pasian, Moncayo, Pagro, Davao de Oro. Here, we explore various innovative practices and facilities dedicated to crop production. These include a well-maintained nursery, a state-of-the-art tissue culture laboratory, and the large-scale production of beneficial biocontrol agents, particularly trichoderma. Now, we're off to our third destination, the Farmer's Field in Maragusan, Magro, Davao de Oro. Here, we visited multiple vegetable farms and had the chance to collect samples of various plant diseases. This visit was not just about observation, it was also about learning and sharing knowledge. We engaged with local farmers and gained valuable insight into their crop management practices. At the same time, we are able to share the knowledge we've gained from our academic courses, creating a meaningful exchange of ideas. As we conclude our visit to the farmer's field, we carry with us deeper understanding of sustainable agriculture and we renewed appreciation of the hard work of our local farmers.
Staining procedure. First, you have to prepare the things you need. On a clean dry slide, paste a loopful of actively growing bacterial culture with the help of wire loop. Apply 1 or 2 drops of gentian violet solution. Keep it for 1 minute. Next, wash with tap water for a few seconds. For 1 minute, cover this film with iodine solution. After rinsing it with water, you can now put the decolorizer until it flows colorlessly from the slide for about 30 seconds. Next, counter stain for about 10 seconds with safranin. Lastly, wash briefly in tap water. In this case, we have identified the bacteria that we cultured is a gram-negative.
Hey guys, I am Brian Intong, a plant pathology student, and for this video, I will show you how to use microscope and to mount specimen.
The first thing you need to do is plug in the microscope and turn on the illuminator. And then use the rheostat to raise the level of the light. You should start with that open halfway. This will then give you light from the illuminator up into the condenser and to your specimen. The next step is to use the coarse focusing knob to lower the stage all the way to the bottom. Open the clip, put your slide on the stage, and release the arm. Use the stage adjustment knobs to move your specimen over the condenser so that it's in the light and centered. Grip the turret to rotate the objectives. Do not pull or push on the objectives. Here is your lowest power objective. This is also referred to as the scanning objective. That's a 5x objective on this scope. Here's the 10x objective. Here's a 40x objective. And here's a 100x objective. Rotate the turret until the lowest power objective is above the specimen. It will click into place. Pull the iris diaphragm lever all the way closed and then slightly open it. When you are looking through the lowest power objective, you will be using the coarse focusing knob. Since you've lowered the stage all the way, you are slowly raising the stage until your specimen comes into focus. When you are in focus with the lowest power objective, use the stage adjustment knobs to center your specimen in the field of view. Use both knobs. As you look through the oculars, one of the knobs will move your field of view horizontally, the other vertically. The stage will move side to side and back and forth. When the specimen is centered and in focus, rotate the turret so that the next highest magnification clicks into place. From this point on, you will only be using the fine focus knob. Using the coarse focus knob could easily drive your slide into the objective and damage both the slide and the scope. Refocus the specimen with the fine focus knob and recenter it in your field of view with the stage adjustment knobs. Once the specimen has been found, focused, and recentered at 40x magnification, the nosepiece should be turned so that the 40x objective and the 100x objective are straddling the specimen. A drop of immersion oil should be placed on top of the slide, immediately above the light source. After the immersion oil is added to the slide, the 100x objective should be moved into place above the specimen. Make certain the 100x is moved into place, not the 40x objective. Oil will damage 40x objectives. Refocus on the specimen using the fine focus knob only, and then recenter the specimen in the field of view. Once done viewing the specimen, the stage should be moved all the way down, the slide should be removed, the immersion oil should be cleaned off of the 100x objective, and then cleaned off of the slide. The lowest objective, 10x for micro, should be moved into place above the light source.
Now, I will show you how to mount a specimen. First is to sterilize the tools using glove. Scrape the specimen and place in slide.
Like, can I circle?